Welcome back, students. In previous sessions, we have discussed about body plans, body structure, body function, embryonic development, and so on and so forth. Now we will dig in more deeper and talk about particular organ systems as it found in the organism, which have organ level of organization, and how these systems function in these animals altogether, as well as what variations do we often find as well. So let's start. In the first part, we'll only talk about organ systems related to structure and sensing of our body, and in later sessions, we'll also cover other systems as well. One of the major components that we see during evolution of animals is the emergence of a head, an area where the nervous tissue can be localized, where the brain can be localized, through which the center of command and of control. So that formed a very important aspect during development of animals. As we can see that have been animals or cases of animals where did not have any head. For example, you see that uh, sponge hair or the sea anemone hair or the starfish hair, you do not see any head formation in that case. You cannot classify something as a head in that case. So this emergence of a head is called as cephalization, where the cephalo word is coming from the reference to head. So from that aspect, the emergence of head or cephalization is a very key component of the emergence of a nervous tissue in the context of brain and spinal cord, if we talk about in that context. However, the animals that are shown on your left side, without any cephalization also have nervous tissue present. But in that case, there is no central command center and the nervous system is more or less distributed. But even if we compare the animals which are non-cephalized or the animals which have show the ability to show cephalization, we still see certain similarities in the structures. We still see certain similarities in the structure of the nervous tissue. So let's dive into that. When we talk about the nervous system, it is composed of two kind of cells, the neurons, also called as the nerve cells, and the glials, which are also called as the supporting cells. Glial cells are further of various types. When we'll discuss in more detail about human physiology in future classes, we can then talk about that in more detail. However, what we can talk about right now is just in the context of how these neurons and glial cells are often organized. There are various terms that are often utilized there and thrown out here and there, but we need to understand what do they actually mean. So neuron or a nerve cell as well as a glial cell is a cell. When various neurons come together, we form what is called as a nerve fiber. And then when various of these nerve fibers come together, we form what is called as a nerve. So nerve cell, nerve fiber, and nerves are connected but distinct terms. This is to understand in just exactly same manner how your wires also are present. There's a huge tuft of wire, then there are certain collection of bundles of wires, and then within that you have individual wires present. It's exactly like just that, because you have to reach to every single cell to give a command and how they have to respond. Now, having said that, these neurons further form masses. Wherever they are present close together, they form collection of masses, especially where they are interacting, where they are bringing a certain signal and sending a certain signal, and these interactions are happening. And a cluster of neurons is often formed along with certain glial cells to support the formation of the chemicals that are required by these cells. This is called as a ganglia. This cluster of cells or a bulbous kind of structure that we see is called as a ganglia. Now, this particular term is often used when we talk about the peripheral nervous system or the nerves which are present away from the central axis and are doing most of the sensory as well as the motor aspect of the responses. However, we call these same tufts as nucleus or nuclei when we talk about the same things in the central nervous system, that is the brain and the spinal cord. The other aspect is when these two ganglia connect with each other. Now, these ganglia may belong to similar responsibilities, or they might have altogether different responsibilities as well. When we see certain nerve fibers connecting two ganglia, two nuclei, which each have similar responses, similar responsibilities, such a connection is called as a commissure. And another connection wherein we see two different ganglia nuclei altogether, we call such kind of signal as a connective, as you can see in the diagram as well. So commissures connect your similar ganglia, while connectives are here to connect your different ganglia altogether. Okay, but these are just terms to explain to you how the nervous system looks like. Another very key aspect is that we have to talk about in the case of animals, because it is incomplete when we talk and speak multiple times about chordates, vertebrates, and all that, is that of a notochord. So notochord is nothing else but a rod-like structure that was found in animals, especially in the embryonic stage. For certain animals, it is retained all throughout their life, whilst in certain animals, it is not retained throughout their life. So in this particular image, that is shown of a tunicate or a urochordate animal, which often shows retrogressive metamorphosis. Retrogressive means reversing back its higher advanced features back to lesser advanced features. So metamorphosis again as such means changing or transformation. So retrogressive metamorphosis is seen in urochordates, uh, group within chordates, which show that its larvae have more advanced features than its adult stage. So the larva does show certain features, but those disappear in the stage of your adult. But that's a different story altogether. Notochord is again an ectodermal derived structure, and this structure helps the animal to provide certain support as well as for certain initial developmental cues that are needed. However, this is overtaken by vertebral column in later animals, and hence you do not see notochord in adult animals and most of the other vertebrates that we will talk about. So now let's come back to what we were discussing. So that way you have animals as chordates and non-chordates. So animals with notochord, chordates; animals without notochord, non-chordate. But then you will say that chordates can also be replaced, can also have a situation or time in their life where they have replaced their notochord with vertebral column or other structure. So yes, but still they have that structure in their embryonic stage, which shows that they are connected. So that is why we call them altogether as chordates. Now, when we talk about nervous system brings appendages, because appendages are something that help us to move around, they help us to sense the world around, they help us to make sense of the world around, and so on and so forth. So there are various appendages such as the tentacles in the jellyfish, or wings or antenna in the case of a moth or a butterfly, the wings and feet of a bird, the parapodia, these segments that are seen in the polychaete annelid, and then we have this tentacles or foot as well as that we see in the case of sensory tentacles also that we see in the case of gastropod, a mollusk. And then we see these fins present in the fishes. Also, at times they have certain specialized structures called as lateral line for sensing the environment. Sharks also have electroreceptory organs. So there are various variations. And these structures are not just helping in movement, but they are also helping them escape whatever response they have to generate to a certain stimuli. For example, a fish uses its fin to finally coordinate to move away from approaching predator. Similarly, the tube feet of the starfish, and this is one of the most beautiful structures that I have obviously seen, because how beautifully they work. From the top of starfish, let's just look like it's not moving at all, but when you look at it, the individual arms, you will see these hundreds of feet working like hydraulic pumps and moving up and down to allow the animal to move entirely. And so that's how exactly the starfish moved. And we have also talked about its friend SpongeBob, but again, another conversation altogether. Let's come back to another feature of animals, that is skeleton. Skeleton could be exoskeleton or endoskeleton, depending on whether the skeleton is present inside or outside of the body. So, not respected because I have said it opposite. So, exoskeleton basically is when the structure is present outside the body, and endoskeleton is when it is present inside the body. So, exoskeleton can take various forms, and most of them are there for protection. It's actually the same for endoskeleton also. It's just that the exoskeleton provides more strength, because in that case, it's even outside of your softer tissues, so that it provides even more strength. But again, that also comes up with own limitation. Think about that. What limitation could come with having a shell outside? Try imagining walking with 10 bags over your head, or wearing a table all over you and then walking around. Imagine how you will feel. That's exactly how these animals with exoskeleton also feel. So these exoskeleton can take a shape of a shell, scales, feathers, hair, cuticles, etc. Carapace also, as seen in the case of a tortoise or a turtle. And again, tortoise and turtle are two different animals. Do not confuse them. The image here is that of a tortoise shell and not of a turtle shell. If you throw a tortoise in the water, it will sink. So please do not do that. Now, coming back to endoskeleton. Endoskeleton is the skeleton which is present internally, and it is derived from mesoderm in contrast to your ectodermal derivative, that is your exoskeleton. This is most of your fibers or spicule kind of structure that we found in sponges, as well as cartilage and bones that is found in most of us animals as well, whether it is your cartilaginous fishes, the sharks, rays, or that is your bony fishes, or other animals, your reptiles, and every other animal which have skeletal bones as well. Now we will talk about the blood vascular system, because we have talked about nerves, we have talked about how they move. But another aspect that we have to understand is how animals move their material around. But we will not dig in too deep into it, because that is more of an area of physiology. But we will try to understand what kind of blood vascular system is seen by and large in animals. This could be closed or open, based on whether they form a complete loop or not. So whether they just leak out the material and then reabsorb it back and then send it back, or whether they are forming proper channels. So basically a contractor that just wanted to do away with the work, and a contractor that was like, I will make the most beautiful and most stringent one, I will connect every single thing together. So just like that, we have open circulatory system and we have closed circulatory system. Open circulatory system often loses out blood in these spaces called as sinuses or lacrinae. The cells are bathed within it, and then the material is again taken up back by another blood vessel, often found in the case of arthropods and mollusks. Then you have your closed blood vascular system, wherein we have proper tubes, the arteries, veins, and capillaries, which form the continuous channels. But again, you do find certain similarities here also, because there are always certain openings which allow certain larger materials to also move out, because at times that is required. For example, a cell has to move out, WBC or the white blood cell has to move out for fighting infections. So yes, there are certain porous nature at certain points, but still by and large it is leak-proof. So this is found in most of your other animals, your higher animals, and they don't empty the content as such usually directly. It's primarily diffusion, the process that is utilized in this particular scenario, as well as certain liquid that oozes out only at the capillary stage, because if any content loses out at any other point of stage, it will lose out a lot of blood. And yes, whenever two these different adaptations arose, they also arose in a very particular context. So you might imagine that we do not see an animal fainting in the case of, say, your open circulatory system, but that can happen in a closed circulatory system because animals' structures are formed in that manner. So they will not receive the requisite nutrient that they want. So as such, you can see that both systems have their own advantages and disadvantages. Now let's end this discussion with a certain question. What is the chemical composition of exoskeleton in arthropod, mollusk, and reptile? Try to see about that. What geological changes might have favored the evolution of closed circulatory systems over open in certain animals? And what advantages do you think an open system solves over your closed circulatory system? Because you do find it in animal, insect animals and in most diverse group. So that we do find out about them.